Welcome viewers. Welcome to the lecture series on prokaryotic gene expression. In today's talk, we are going to discuss about prokaryotic translation. The genetic information which is encrypted within the genome in the form of DNA gets converted into RNA by a process called transcription. The second stage of gene expression is called translation, wherein the genetic information which is embedded within the mRNA in the form of ribonucleotides gets converted into a polymer of amino acids. Translation is supposed to be one of the most highly conserved processes among all living organisms, and it is also one of the most energy demanding steps in all living organisms. If we consider the energy demands, then we will see that 80% of the cell's energy is being utilized in carrying on the process of translation, and almost 50% of the cell's dry weight is involved in translation. It will be astonishing to understand that for synthesis of single protein, we need the involvement of 100 different proteins. So if we consider the process of translation, it comprises of 4 important components. In today's lecture, we will be discussing about these 4 important components. So the component 1 is called messenger RNA or mRNA. The second component is called transfer RNA or tRNA. And the third component is the enzyme called amino-acyl tRNA synthetase, which is involved in charging of the tRNA with a proper amino acid. And finally, the submicroscopic particle called ribosome, that is catalyzing the process of peptide bond formation. So in all, we will talk about all these 4 different components. So before getting into the details of these 4 different components, let's have a look onto the eminent scientists and their contributions towards development of translation. The first name that comes in our mind is Francis Crick, who first coined the term central dogma, and he was the one who thought that if the message which is present within an mRNA is present in the form of ribonucleotides, unlike the process of transcription, translation is much more challenging. Because in transcription, we can see that the information which was present in the form of deoxyribonucleotides got converted into ribonucleotides. But still, the nature of the building block was same. That is, both of them were nucleic acids. But in case of translation, we will see for the first time that the information which is embedded in the mRNA in the form of ribonucleotides is getting converted into a polymer of amino acids. So there is a changeover between nucleic acid to amino acid. And what else? We have also seen that there is no such complementarity between the side chains of amino acids and the purine and purine bases of the mRNA.
Ma'am, if there is no complementarity between the amino acids and the mRNA, how is the message of the mRNA getting translated to the amino acid?
Yes, this was supposed to be the most challenging question which was raised by Francis Crick, and he proposed that there should be some adapter molecule present that should be capable of interacting with the mRNA, as well as it should be interacting with the amino acids which are getting polymerized to form the protein. So initially people were thinking of, or Francis Crick was proposing, that the adapter molecule should be of RNA in nature. But he was unable to establish the exact nature of the RNA. It was a contribution of Paul C. Jaminik and Mahlon Hoagland. These 2 workers, after 2 years of Francis' proposition, Jaminik and Hoagland finally established that yes, it is an RNA, and they named it transfer RNA. And it comprises of 15% of the total RNA population inside a cell. They called it transfer RNA because this transfer RNA was capable of interacting with the nucleotides of the mRNA, and the amino acids which were attached to the tRNA were transferred during the peptide bond formation. There came the name transfer. So now we finally start up discussing about all these 4 different components one by one. Let's start up with mRNA or messenger RNA. The bases in the mRNA are read off in a group of 3 nucleotides. These 3 nucleotides are called as codons. Now the question arises, does the translation is done on the entire mRNA? No, there are certain regions in the mRNA which are not getting translated. They mostly lie in the upstream region. So translation starts from the 5' end. In the 5' end we do have untranslated regions called 5' UTR. That doesn't get translated. So translation actually gets initiated with a codon called start codon and it ends up with stop codon. So the sequence of nucleotides or ribonucleotides which are lying between the start codon and the stop codon is called as an open reading frame or ORF. Now the question arises that during the decoding of the message which is encrypted within the mRNA, the foremost requirement is the loading of the ribosome onto the mRNA.
Ma'am, I have a question. During transcription, there is a sequence called promoter sequence that helps in the recognition. But during translation, whether such sequences are present or not, that helps the ribosome to recognize the mRNA and binds to it.
Exactly. Here in case of mRNA also, we find certain recognition sequences, and as in transcription, the recognition elements never get transcribed. Here also, the recognition element is not getting translated, and that recognition element is called as ribosome binding site or Shine-Dalgarno sequences. These sequences mainly comprises of AGG nucleotides. These sequences are recognized by the ribosome, and RNA component of ribosome called 16S rRNA component of ribosome specifically comprises of CCU sequences that forms hydrogen bonds with this Shine-Dalgarno sequences of the mRNA. So more is the conservation of this Shine-Dalgarno sequences, better is the interaction between the Shine-Dalgarno sequence of the mRNA and the rRNA component of 16S rRNA of ribosome, and the spacing between this Shine-Dalgarno sequence and the start codons. These two factors actually decide or dictate the fate of translation. So better is the distance between the two, better is the homology, better will be the translation rate. The thing that comes in our mind, that how the decoding of the nucleotides is to be done into amino acids. So we have got chosen 3 nucleotides and we are calling them as codons that code for 1 amino acid. So out of 4 nucleotides, if we take 3 at a time, then we can have 64 different probabilities of codons, out of which 3 codons are dedicated as stop codons. It means we have got 61 codons which code for amino acids, but we have got only 20 amino acids to code. Then obviously 1 amino acid will be coded by more than 1 codon. Now the question arises, if 1 amino acid is coded by more than 1 codons, then who is going to recognize these codons? As we have said that the recognition is done by the tRNA molecules, and the region of the tRNA molecule that carries on the recognition is called as the anticodon region. It means if we have got 6 codons for serine, say for example, then we need at least 6 different tRNAs with 6 different anticodons, which is energetically very much demanding. As I said, translation is already overburdened with demand of energy. So to minimize the wastage of energy, the system opts for a new option wherein one tRNA can recognize more than 1 codons. Then how it is made possible? It is made possible by the hypothesis called Wubble hypothesis. This hypothesis was again being launched by the famous person Francis Crick. He said that In a mRNA which is read in 5' to 3' directions, and the tRNA have got anticodons, then the interaction between the first two nucleotides of the mRNA with the tRNA is perfectly Watson-Crick base pairing. On the other hand, the base pairing with the third nucleotide of the mRNA and the first nucleotide of the tRNA is non-Watson-Crick type base pairings. So, we can see here that the first two nucleotides of the mRNA that form hydrogen bonds with the two nucleotides in the tRNA are perfect Watson-Crick base pairing, but the third nucleotide of the mRNA is interacting with the first nucleotide of the tRNA in some different type of interaction which is other than Watson-Crick base pairing. Now we will talk about the second important component of translation machinery, that is the tRNA. tRNA contains almost 80 nucleotides. It has got 4 stem loop structures and 3 loops. It means tRNA has got 4 short base paired stems and 3 loops. Different tRNA molecules vary considerably in sequences. The acceptor stem of the tRNA is made up of base pairing between the 5' end and the 3' end of the tRNA. The 5' end contains almost always a G residue, and the 3' end of the tRNA ends up with a single-stranded nature of CCA residues. The amino acids which are attached to the tRNA molecules are attached to this 3' end, and the attachment is done utilizing the carbonyl carbon of the amino acid and the OH group of the adenine residue which is present at the terminal end of the tRNA. The anticodon loop which is present halfway around the sequence in the anticodon loop. It lies in between the anticodon loop. The anticodon is always preceded by 2 pyrimidines, usually uracil, and is followed by a modified purine. In case of tRNA, for the first time we will see the incorporation of certain unusual bases. As we have seen that there is incorporation of thymine which is not present in case of RNA. We will see the incorporation of pseudouridines. We will see the incorporation of various unusual bases that like hydrouracil is there. Then we have got various quinosin and many other strange bases are there like methyl uracil, and they actually help in the proper folding of the tRNA. These unusual bases in the tRNA help in the recognition of the anticodons with the codons on the mRNA. So, this unusual bases really play a vital role in functioning of the tRNA. The third important component of the translation machinery, that is the amino-acyl tRNA synthetase. Amino-acyl tRNA synthetase is the enzyme that carries on the charging of the tRNA. Charging of the tRNA means loading of the tRNA with an appropriate amino acid. So, the enzyme amino-acyl tRNA synthetase really faces two important challenges. The first challenge that it faces is to incorporate a proper amino acid onto a proper tRNA, and the second challenge is that it should recognize the correct tRNA. The reason behind is that amino-acyl tRNA synthetase has to select among the iso-accepting tRNAs. As for example, we all know that for a particular amino acid, we can have more than one codons. It means we have got more than one anticodon bearing different tRNA molecules that are to be charged with same amino acids.
Ma'am, the question is,
Yeah.
how does the amino acid synthetase molecule recognizes the particular amino acid has to go with the particular tRNA molecule?
Yeah, the question is very nice, that tRNA should be recognized properly by the amino acyl tRNA synthetases, as it should have certain elements within the tRNA which are present in common in all the tRNAs that code for the same amino acid. So we will see here that obviously that recognizing element is not present within the anticodon region of the tRNA. There should be certain recognizing elements present there. So we call those recognizing elements present within the tRNA as called discriminator sequences, or which are present in the acceptor stem and the anticodon loop. They too actually help the amino acyl tRNA to recognize the correct tRNA. Next important element is called as the anticodon loop, that also helps in recognition of the proper tRNA. So these two elements are sometimes called as the second genetic code because they help in recognition of the proper tRNA. Next is, during charging of the tRNA with an amino acid, it is basically done in two steps. So let's get into the chemistry behind the charging of tRNA. So the first step of charging of a tRNA with an amino acid is called as adenylation. In this step, the amino acid interacts with one molecule of ATP, leading to synthesis of an adenylated amino acid and release of a pyrophosphate molecule. This is a first step wherein one molecule of ATP is being utilized. And the next step of translation or charging of tRNA is the addition of proper tRNA molecule with the amino acid. Here we will see that this adenylated amino acid is being attacked by the lone pair of electrons on the OH group of the tRNA, attacks the carbonyl carbon, and which leads to charging of the tRNA with a proper amino acid and release of AMP molecule. This bond is a very high energy bond, that is the bond between the amino acid and the nucleotides of the tRNA. Why we are focusing on the energy status of this bond? Because during peptide bond formation, there is cleavage of this bond leading to formation of a peptide bond. And therein we will see that while forming peptide bonds, we don't need any energy. That energy which is obtained by cleavage of this high energy bond is being utilized in making the peptide bond. So this is very much important step from the viewpoint of making the platform ready for the translation to carry on. Next, we will talk about that how that amino acyl tRNA synthetase actually recognizes that it has incorporated a proper amino acid onto a proper tRNA. If there is some sort of mistake in addition of the amino acid, then it will lead to misincorporation of amino acids during translation process. That's why we have seen that this amino acyl tRNA synthetases do have two pockets. One is called as the catalytic pocket and the second is called as the editing pocket. Let's take up the example of isoleucinyl tRNA synthetase. Isoleucinyl tRNA synthetase is responsible for loading of isoleucine onto its corresponding tRNA. So if I compare the structures of isoleucine and valine, then we will see that there is a difference of only one methylene group. Then what we will see, if I compare the incorporation rate of valine and isoleucine onto the isoleucinyl tRNA synthetase, then both valine can also get entry into the catalytic pocket of isoleucinyl tRNA, and isoleucine should obviously get entry into the catalytic pocket. Then how the system realizes the difference between isoleucine and valine? We will see that this extra methylene group in isoleucine gives it an extra stable interaction, and this extra stability is responsible for isoleucine getting more access onto the catalytic pocket. But this leads to a error rate of 1 in 100, but we have seen that the error rate is pretty less than it. So almost among 10,000 amino acids that are incorporated, there is misincorporation of only one amino acid. So obviously there is lying some another step of proofreading, wherein if certain mistakes are being incorporated, they are nullified there, and that proofreading pocket is called as the editing pocket. So as I said, that valine is capable of entry into the catalytic pocket, and out of 100 such cases, in one case there is chance of misincorporation, but the story doesn't end there. What happens that this wrongly incorporated valine has to pass the catalytic, after catalytic pocket, it has to pass on to the editing pocket. And the size of the editing pocket is made so that all the amino acids which are smaller than isoleucine can get entry into that pocket. Because the tRNA synthetase is meant for isoleucine, so isoleucine will never get entry into that editing pocket. But all the shorter amino acids that got charged because of mistake gets entry into that editing pocket, and they are getting hydrolyzed. That it means if the mistake is done during the catalytic pocket, then these mistakes are being taken care of in the editing pocket. Finally, we will talk about ribosomes, which is the decoding machinery of the translation apparatus. Ribosome mainly comprises of rRNA and proteins. So the sub-organelles which actually make ribosomes were first studied by ultracentrifugation of the bacterial ribosomes, which leads to separation of the components. So prokaryotic ribosome is mentioned as 70S, where S stands for Swedberg units, means the sedimentation coefficient. Next is, if we precisely look into the components that actually make the 70S ribosomal subunit, it comprises of one larger ribosomal subunit called 50S and a small ribosomal subunit called 30S. This 50S can further be divided into 5S rRNA and 23S rRNA plus 34 proteins. And if we look into the smaller ribosomal subunit 30S, it is made up of 16S rRNA and almost 21 proteins. This larger and smaller ribosomal subunits of the ribosome are attached to the mRNA utilizing the Shine-Dalgarno sequences of the mRNA, and they carry on the translation of the proteins. Now, the question arises that in prokaryotes, transcription and translation are coupled, and the single mRNA that is undergoing translation is being translated by more than one ribosomes together. It means the single mRNA, when carries on translation more than once, then there is attachment of multiple ribosomes onto one mRNA. Such case is called as a polyribosome. Now we will talk about the steps of peptide bond formation during protein synthesis. So if we focus onto the ribosome, it has got three sites called P site, A site and E site, where P site comprises of peptidyl tRNA binding site and A site comprises of amino-acyl tRNA binding site, and E site is called as the exit channel or exit site. And what happens during peptide bond formation is that we will see that there is attack of amino group of the amino acid that is attached onto the amino-acyl tRNA, which is bound onto the A site. It attacks the carbonyl carbon of the amino acid that is attached to the peptidyl tRNA. This leads to the transfer of the entire peptide chain from the peptidyl tRNA, which is bound on the P site, to the amino acid tRNA, which is bound onto the A site, leading to movement of the ribosome.
Ma'am, which component of the RNA is responsible for the peptide bond formation?
Okay, this is a very good question, and it took a large amount of time by the scientists to prove that which component is responsible for carrying on the peptide bond formation. Usually people used to think that the protein component of the ribosome is responsible for peptide bond formation, but later on it was found that it is the 23S rRNA component of the larger ribosomal subunit that has got the peptidyl transferase activity in it, and it is responsible for carrying on the peptide bond formation. Now the question arises that how it was established. So people found that the catalytic region where actually the peptide bond formation takes place, it is mainly made up of rRNA components, and the protein residues of the ribosome are lying onto the periphery. And what is the purpose of these proteins that are in peptide bond formation? They could find that they are responsible in shielding the negative charge repulsion between the sugar phosphate residues of the 23S rRNA. And they provide the 23S rRNA with a proper conformation so that the amino acids which are bound onto the A site and which are bound onto the P site can be brought into a close proximity to facilitate the peptide bond formation. But the actual key component or actual catalytic activity is being shown by the 23S rRNA component and not the proteins. So this was the first time wherein we could see that RNA can have catalytic activity. It is acting as an enzyme, and hence ribosome is called as a ribozyme.